How's it going? So my name is Dustin, and I'm going to be making videos that should be valuable to people who study either cancer biology, cell death, and cell differentiation. To get the most out of the videos, you should really be familiar with the hallmarks of cancer, or have taken cancer biology at some point. The first video is going to be aimed at really shifting your overall perception of cancer and how you internalize new information relevant to this disease. I do have behind the camera a type up of some of the things I do want to cover, so if it does look like I'm reading a few things, that is just to make sure I'm keeping myself on track and I'm able to convey the information in a way that is effective, digestible, and easy to understand. So, who am I? No one interesting, really. I've had something of an on-again, off-again obsession with cancer for about 10 years now, which is when I took cancer biology. Basically, back then, I'd grown more than a little bit obsessed with the science and have since spent thousands of hours studying the intracellular regulation that is relevant to cancer signaling, and that entails exploring the literature, right? This isn't textbook understanding. Thousands of articles, and not so much just reading, but also ensuring I was taking notes, trying to piece together cross connections between these signaling networks so I could have a better understanding of what's going on. I'm hoping that with these videos, I'll be able to outline to you some of my perspectives on how these signaling frameworks work, and the fact that this is going to be a public forum is going to be extremely valuable. I, more than anything, would love to have people to talk to about this information. The public forum gives you an opportunity to keep me honest, correct me if I'm wrong, and, we can have a little bit of fun. So, the first video here is going to be aimed at changing your conceptualization of cancer, and it's mostly going to be talking about broad strokes events. The second video is going to provide some of the background molecular mechanisms that underpin the discussions. It's going to explore cell death signaling, NF-kappa B signaling, proliferative signaling, and DNA damage response signaling, and how some of those are going to be coordinated with differentiation. Let's see here. The second video is probably going to be a little bit more lecture-esque. I have already made the PowerPoint for it. It is about 80 slides and is rather dense. That video will be a little bit better to be actually studied, because you want to make sure you have that background framework for understanding the cross connections between these signaling networks before we get to videos 3 and onward, which will be exploring new networks as I integrate them into my understanding of how cell biology is working. So, just to kind of give you an example of what these discussions are going to look like, I do want for them to be high level. So when we're looking at, for example, what I'm kind of studying now is mitochondrial associated ER membranes and their reciprocal regulation by cell death mediators in the control of calcium oscillations. So calcium exchanges can be regulated by low-level cytochrome C release, and it can bind to the inositol triphosphate receptor at the endoplasmic reticulum and stimulate further calcium release. However, high concentrations of cytochrome C can have the opposite effect. So mitochondria can accumulate calcium to activate mitochondrial calpain, which can cleave and facilitate the release of AIF into the cytoplasm, and this is positioned to control DNA, the DNA damage side of the apoptotic signaling network. And it's important to note that AIF has oxidoreductase signaling activity and is important for maintaining the stability of the electron transport chain. So, in being able to we can see how calcium signaling is positioned to also modify energy metabolism. It is worth noting that calpains can also cleave and prevent the activation of caspases, so we can kind of envision a means of mobilization of specific components of the apoptotic network through calcium signaling. Kind of extending beyond that, and what I'm looking at is, it is worth noting that the reduced version of cytochrome C is not sufficient to generate the formation of the apoptosome. So if you're getting it following cytochrome C release from the mitochondria, if it is either in the reduced form or subsequently reduced, which may be mediated by an enzyme that is localized to the ER membrane, that can prevent the apoptosome forming and facilitate cytochrome C being able to promote calcium signaling in the absence of death. It's worth noting that, of course, phosphorylation of APAF1 by RISC, the ribosomal S6 kinase, which occurs downstream of ERK and mitogen and MAP kinase proliferative signaling, can restrict APAF1 from assembling into the apoptosome too. So this is sensible as mitogenic signaling often converges on calcium release. You've got the proliferative impulse and the restriction of death, while this cell death mediator is facilitating the proliferative side of the actual signaling cascade, or has the potential to. It's always very difficult to say if this is happening in all of the cells, given that the specific cell types in which case these things are discovered might not be recapitulated in all of the different types of cells that we study. Adding on to this, of course, DIRP1, dimer-related protein 1, which promotes mitochondrial fission, initiates fission by recruitment of the mitochondria to the ER membrane, at least by one mechanism. So there's a lot of far-reaching implications for this, right? Given the potential impact of altered fission-fusion dynamics on energy metabolism, and the fact that fragmented mitochondrial morphology will often correlate with aggressiveness and lower survival rates in cancer. So we can kind of see how through calcium signaling, you can have this combination of proliferation, calcium, and death mediators all intersecting at these mitochondrial-associated ER membranes. And these localizations are going to be really important for regulating differentiation, I'm pretty sure. So generally with these videos like 3 and on, we're going to be looking for exclusionary signaling events that can kind of give us insight into the activity of the broader system as a whole. Most signaling events are biphasic, with a primary step preceding an execution step, and we're going to be trying to find out what happens in the interim signaling events that allow the cell to sense and ensure that the environment is appropriate for the execution step of these events. Now, I know that this is not the best forum to actually have these discussions, just me speaking into a camera, so I do intend to get a projector and a whiteboard so we can have a little bit more, well, fun and give you some visuals that'll be able to make it clear on what's going on. But back to the video at hand and my above stated purpose. I want to change how you conceptualize cancer and help you integrate an understanding of several hallmarks into a conceptual framework that allows you to see the interrelatedness between death, differentiation, and cancer itself. So there's a couple things we're going to need to discuss here, and a lot of these were not something I was introduced to when I was going through my studies, and some of them are of my own interpretation. So one of the main ones is cell death and differentiation. We all know that, yes, it's a journal. I did not, and I don't think a lot of people have a full appreciation for how interrelated these signaling events are. It's absurd, honestly. It was proposed back in 2007 that apoptosis is really just cellular differentiation taken to the extreme. It's a bit of a simplistic outlook, but it's not really wrong in a lot of ways. So basically the protein mediators of cell death, your death receptors, FAS, tumor necrosis factor receptor, your death receptors 4 or 5, aka trail receptors, caspases, calpanes, BCL2 family members, inhibitors of apoptosis, others, like all of them are able to control differentiation, and frequently by performing canonical functions that are typically associated with cell death. Now, how is this controlled? I mean, you've got your basic mechanisms that are proposed, right? Like attenuated levels of protein activation, spatiotemporal control, shuttling them in and out of this location. And of course, alter transcriptional and translational regulation, but that's not very compelling because, I mean, really, isn't everything regulated like that? It is worth noting that these proteins can actually mediate differentiation by performing functions that are typically canonical and associated with apoptosis. One of the, unfortunately, best characterized interactions is going to involve caspase-3 cleaving ICAD, inhibitor of a caspase-activated DNase, to facilitate CAD's translocation into the nucleus. In this, occurs during myoblast differentiation, specifically. This system sees CAD translocate to the P21 promoter, induce a single-strand DNA damage break, as opposed to a double-strand one you would typically associate with apoptosis, and this induces P21 to halt cell cycle and facilitate cell cycle exit associated with terminal differentiation. XRCC1, mediated repair of this damage, is critical to proper differentiation, and in video 2 we're going to go into a little bit more detail here, but it is about not just this particular event, but a variety of the signaling events that have been either directly linked to or associated with differentiation. But it is worth noting that it is well established that the mediators of differentiation regulate cell death. I'll link a couple articles in the bottom just so you can have that clarity, but it's super important. And based on this, there are a couple extremely important takeaways. One is kind of like the baseline beauty of the system, right? In utilizing cell death mediators to regulate differentiation, dysregulation of the system should lead to either death or senescence. And this can work as a built-in safeguard to protect multicellular life from cancer transformation. So it's really cool. But realistically, a more important takeaway is going to come from the fact that a lot of the research that we've done, especially on like cell death, looks at interactions and infers from or interprets the interactions on the basis of assumption that cell death is the intended outcome. Sometimes it is, but it's not necessarily going to be the case in a lot of these circumstances. We need to kind of take a step back, remove that presumption, and look at the core mechanics of what is actually going on in these cells. It gives us the ability to reevaluate, and it gives us an abundance of information on how these signaling networks interconnect. So, let's discuss why the duality of function in cell death mediators makes a lot of sense, at least in my mind. We often conceptualize cancer as cell specialization, and it is, of course, but I don't really think that's the best way to think about it. I think it's better aligned when we look at how the cells are sensing and integrating these signals to view cancer as an adaptive stress response that entails proliferation. And this is one of the reasons NF-kappa B signaling is going to be so important. But consider also the utility of using these proteins to mediate differentiation. When we look at all these different cell types, the phenotypic variation is extraordinary. I mean, it really is. Caspases and some of these proteins can mediate all of that. Enucleation, that's fine. Cytoskeletal reorganization, caspases can do that. Motility, fine. Like, they can regulate everything. So the way I see it is it's really going to be about flexing distinct arms of the cell death signaling network during differentiation of these cells. We need to understand how that regulation is controlled so we can have a better appreciation for, well, just mechanisms that govern differentiation as a whole. Now, I have to say, like, I'm not really convinced that this is just limited to apoptosis, right? The number of cell death pathways at this point is insane. Well, not really, but there's a lot. You've got apoptosis, necroptosis, pheroptosis, parthenatose, netosis, cuproptosis, and I'm sure there's plenty that I'm not even thinking about. Now, some of these versions of cell death, of course, are going to be intended to kill this cell. These pathways are meant for that purpose. And I think you're really going to see that a lot more when we're looking at immune system pathways, right? Netosis, where the neutrophils weaponize the DNA to fight infection during death, which is really cool. The way I like to think of it is that these forms of cell death are simply failures to accommodate stresses that the cell is subjected to. Some of them may be intended or have the potential to facilitate differentiation based on the surrounding signaling context and what the environment is subjecting the cell to. So I don't think it's just apoptosis and those signaling networks. I think it's very broad, but there's a lot more work to do in outlining how that could possibly work. Cool. So moving on from here, we really need to have a discussion about DNA damage. DNA damage gets a really bad reputation, right? And this is understandable as it can potentially be mutagenic, right? But DNA damage is central across lineages to regulating cell differentiation. I mean, ubiquitous? I don't know, but I would not be surprised if it was, and I think it probably is very close. So we actually just had a conversation here, right, about how regulated DNA damage can facilitate differentiation through the induction of P21 and the exit from cell cycle during myoblast differentiation. Now, it's also important to note that other types of cells, stem cells especially, utilize DNA damage to regulate their differentiation as well. Hematopoietic stem cells do, embryonic stem cells. You'll see B cells maturation, T cells, of course, when you get VDJ recombination. This is interesting and can make some sense, especially if we're talking about damage that's induced in G1. Because, at least in early G1, you tend to see heightened levels of chromatin plasticity, or at least according to one article I read. So when we think about the chromatin architecture in the nucleus and the degree of remodeling that would need to take place to facilitate the changes in transcriptional profile, it would make sense if we have nucleases that are able to instigate and facilitate this remodeling by recruitment of DNA damage response agents and epigenetic regulators that facilitate that process. It is worth noting that CAD, caspase-activated DNase, induces DNA damage during apoptosis in a biphasic signaling manner, at least biphasic. It's typically considered to translocate to the nucleus, localize to the nuclear periphery, and induce DNA damage and cleave these 50 to 300 kilobase structures from the periphery. Now, we do know that heterochromatin in most cell types is localized to the periphery, at least generally. So if we're thinking about changes in transcriptional profile, it makes a lot of sense, at least to me, that we could be utilizing this signaling event to modify that architecture so that the following cell cycle exit, the cells are able to have reordered the structure of their chromatin such that they can induce the appropriate genes necessary for the new cell type. Let's see here. Cool. All right. Now, let's talk about some other nucleases, right? It's most likely not going to, there's no way it's just CAD, right? There's far too many nucleases. Endonuclease G, which can be released from the mitochondrial intermembrane space during apoptosis, has shown to be involved in B-cell class recombination. MIF, macrophage migration inhibitory factor, I believe, is widely expressed during embryonic development, and its expression does coincide with organogenesis. I'm not sure how many of the nucleases are involved in this, but I would expect that a lot of them, and it really does need further research. One of the things that anyone who has started signaling and has an appreciation for the redundancy in these networks can appreciate that in the cell, if it happens twice, or at least as I think of it, if it happens twice, it happens 100 times, just in slightly different manners. So if we've seen CAD do this in this context, we've seen endonuclease G facilitating B-cell class switch recombination, we've seen the correlation between MIF regulating differentiation during embryonic development. The likelihood is it's going to be other nucleases involved, and it will be cell type specific in which proteins get activated and mobilized to facilitate differentiation for that type. Okay, and actually, getting back to the release of endonuclease G, I do personally believe that selective substrate release from the mitochondrial intermembrane space is going to be core to differentiation as a whole. We'll see if I'm right. This kind of brings in another thing into question, right? When we look at if we look at the potential release of proteins from the mitochondrial intermembrane space regulating differentiation, right? That has to, by necessity, lead us to consider what are the distinctions in type 1 versus type 2 apoptotic cells. Type 1 basically do not require the mitochondrial amplification loop to undergo cell death in response to, I believe it was death receptor activations, but I'd have to double check that. Type 2 cells do. So when we're looking at differentiation requirements and the potential for selective substrate release from the mitochondrial intermembrane space to be regulating this, it's going to be important to consider which of those cell types we're looking at, and whether the type 1 or type 2 cell types, and what potential alterations in the regulatory requirements there are to understand how those cells differentiate. Now, so cell death mediators are core components in regulating differentiation, and they can do so by performing some canonical functions, including via enzymatically induced DNA damage. But remember that DNA damage is inherently cell cycle regulated, such that phase-specific activations of DNA damage checkpoints occur in most instances of DNA damage. For example, like in contrast to myoblast differentiation above, where we spoke about DNA damage being induced in G1, and that led to the accumulation of P21 and cell cycle exit, P21 nuclear accumulation is restricted in S phase, and it can lead to re-replication. I believe it's CLR4 with CDT2 as a component of that complex that mediates the degradation of P21 in this instance. Cool. So it's going to be important to be mindful of these distinctions in the cell cycle in the responses to DNA damage on the basis of which cell cycle it occurs in. Now let's kind of discuss proliferation. When we're looking at a basic system, especially in terms of differentiation, we need to consider, at minimum, 3 passages through cell cycle. So if we look at consider a stem cell that is housed in a hypoxic niche, as they usually are, that is signaled to differentiate, it is going to proliferate first. One of the daughter cells is going to alter its specialization, and one of the daughter cells is going to stay a stem cell, so that the population can be maintained. Following that, the more specialized cell should continue through cell cycle as a transit amplifying cell. And there, of course, could be multiple instances where progressive differentiation occurs during the clonal expansion of these cell populations. Now, it's important to understand that we can't assume that the regulation of progression through these passages are going to be the same. The initial passage and the subsequent passage, they should be different. And the final passage also needs to be considered separately as well, after which you would really expect to see stabilization of the daughter cells in about a hypophosphorylated PRB prior to exiting cell cycle. So there's really a lot of considerations here, right? So yeah, there's a lot of considerations here, especially when we're considering stem cells, stem cell metabolism, and cell cycle interactions and regulation you might anticipate being different in stem cells versus differentiation cells that are differentiated cells that are induced to proliferate, or transit amplifying cells. For one of the things that I'm kind of thinking about here is normally when cells approach the G1S transition, they dramatically increase mitochondrial fusion, hyperfusion really, to the point where you basically create this massive tubular electrically continuous network of mitochondria with upregulated ATP generation, mitochondrial membrane potential, etc., that facilitates the energetic demands of the cell as it is going through this process. But if we think about that in terms of a stem cell, in a cell that is in a hypoxic niche, that is induced to proliferate and undergo that first passage through cell cycle, would it do this? Right? That's a lot of oxygen consumption for cells that are very invested in keeping reactive oxygen species low in their cells, maintaining sufficient buffers and redox regulation or systems like your glutathione and thyroredoxin systems. I'm not so sure it would work the same in those cells, and I think that would be an interesting thing to look at. And I can't help but wonder, is it survivin that mediates progression through G1 in stem cells in that first passage? It would make a lot of sense, given that, well, next video. Cool. Now we kind of need to step back and talk about cross-passage regulation of cell cycle. We can't look at these passages individually because there is information communicated from the mother and possibly the mother's mother cell to the daughter cells. So in G2, or rather going back to G1, we know that commitment to cell cycle progression occurs after bypass of the restriction point and hyperphosphorylation of PRB by the cyclin-dependent CDK2 cyclin E complexes, right? But continued progression through cell cycle is regulated in the G2 phase of the mother cell, at least in part. What you basically get is mitogenic signaling in G2 leads to an upregulation of cyclin D1, which is downregulated in S phase, and this happens downstream of ERK signaling and will alter the proliferative environment of cells after they have passed through mitosis. Basically, you can get a couple of distinct populations of cells, one being cells that have elevated cyclin-dependent kinase 2 activity, lower levels of the CKI P21, and a shortened G1. The other population has, relatively speaking, lower levels of CDK2 activity, elevated P1, and a lengthened G1, G0 phase. Now we would expect that the lengthened G1 and G0, G1 phase cells are more primed to exit cell cycle, but the mechanisms of that are not really clear at this point. So beyond the ability of signaling events in G2 of the mother cell to be able to mediate cross-passage cell cycle regulation. We also have to consider that DNA damage in S phase of a mother cell can also regulate the following G1. Some cells will basically accumulate DNA damage during the regular, unperturbed passage through cell cycle, and these lesions will escape repair and lead to the formation of 53 binding protein foci in the subsequent G1 associated with cell cycle arrest and hopefully repair. This is really interesting because we did just talk about DNA damage downstream of CAD mediating cell cycle arrest to facilitate cell cycle exit during the process of terminal differentiation. It would be interesting to look at mechanisms, especially the interplay with 53 binding protein and BRCA1, to see how cells might bypass DNA damage checkpoints to allow for this to happen, such that you get G1 arrest in at least one of the two cells in the following cycle, and how that might coordinate with cell cycle exit and differentiation. So, my current working theory, and this has a lot to be proven, is that we really shouldn't be looking at DNA damage checkpoints as just response systems to deleterious DNA damage lesions as cell cycles progress through cell cycle. Well, as cells progress through cell cycle. I think of these more as most likely communication nodes, whereby you would see transient instances of DNA damage being used to communicate information, well, sense and communicate information that is propagated to, well, is either maintained in that cell or propagated to new cells, such that you would get altered epigenetic regulation and differential DNA damage checkpoint responses as the cell continues to go through cell cycle. I mean, we do see, of course, and this kind of aligns, right, that p53 is normally going to be activated in transient bursts through progression through cell cycle. If these are in response to normal enzymatically induced DNA damage and DNA damage responses, that would make a lot of sense to me as far as how this can be controlled. Now, there are a lot of mechanisms of DNA damage bypass that have been reviewed. I'll link an article in the below, the same, only different, and the subtitle is very long, but it's a great article. Exploring those is going to be something I will probably be doing in videos following 3 at some point. Well, I've explored them, but outlining them in a video context so that we have the opportunity to have a discussion on them is something I do plan to do in the future. So let's kind of recap real quick. And I know this is probably a lot of new information. I'm not giving you a lot of examples yet. That'll be video 2. But cell death mediators regulate differentiation. Not all of them in all cell types. Different arms can be activated based on the needs of the cell as it differentiates. But this is core, this is common, this is established. This is something we need to make sure we integrate into our understanding and perception of what is going on in the cell. Enzymatically induced DNA damage, well established, right? This is core to differentiation, not fully understood right now, which is very unfortunate. And we know that enzymatically induced DNA damage is cell cycle dependent, such that you get specific responses based on the phase in which case it is induced. So we need to be looking at how these different types of damage elicit distinct responses in different cell types, and what the downstream consequences of those are going to be in both regular cells and, of course, cancer cells. So let's actually talk cancer, right? I view cancer as a failure of cell differentiation. Now, I am very much aware, right? Cancer is not something you can just study in the context of individual cells. it is at minimum a tissue level and probably an organismal disease, right? I know that you've got your heterogeneous populations of cells, immune system cell infiltration, signaling events between cancers and the tumor-associated stroma that can facilitate the progression of the disease, especially depending on what those reciprocal interactions and signaling events are. But if we look at, or I would rather say it's just more valuable conceptually to think of cancer cells as cells that are undergoing a defective differentiation program. And it's much more useful, right? We already know that there's variability in how well or how poorly differentiated cancer cells are. And the poorer differentiated they are, the worse the outcome in most circumstances. So, to really kind of slot the framework of differentiation in cancer, we can look at the three-passage basic system, right? So we know that a variety of tumors have cancer stem cells that can repopulate the tumor mass, even if you're able to eliminate some of the more differentiated cells. So when we're thinking about the three-passage system, we would expect that their differentiation is going to look, their proliferation is going to look a little bit more like that first passage. It's not going to be quite the same. We would expect that most of the cells in cancer are going to be behaving like the second passage, such that they are akin to transit amplifying cells that are unable to discontinue their cell cycle progression and terminally differentiate. So it's going to be due, of course, to the characteristic mutations you're going to see in cancer that drive that hyperproliferative phenotype and, well, a variety of other things. Now, we also have to have an appreciation that stem cells are held in hypoxic niches for a reason, right? It's important for their metabolism, their survival, their ability to maintain genomic stability. So when we think about cancer cells, we know that there's variance in the amount of oxygen that's going to be available to cells in these masses in tumors that have solid masses, right? Because the vasculature is not formed correctly, as we know, and that's going to cause issues with oxygen diffusion limits in those particular masses themselves. So the variation we're going to see in cancer and the spectrum of phenotypes will be more complex than we're able to really describe in a three-passage system, but it still gives us a good conceptual framework to understand this larger system of differentiation as a whole. All right. So let's consider some of the hallmarks of cancer and how they relate to properly regulated differentiation, right? So, replicative immortality. Differentiated cells normally express telomerase, right? I mean, obviously the alt mechanisms employed by cancer are interesting. I've not spent enough time with them to be able to speak on those in this moment, but if we think about a healthy system, stem cells can express telomerase, no big deal. Genomic instability, right? This one's interesting because regulated DNA damage controls differentiation. We don't really have a full outline of how widespread that is, but it really makes sense considering cancer cells self-inflict DNA damage to promote their survival. And I literally found this out today. This can be done by caspase-activated DNase in G2, not G1. And it's not, at least what I initially read, is not dependent on caspase activity, which is it's perfect. We're starting to see that these same proteins are functioning in similar capacities in multiple cell cycle phases to coordinate this process. Obviously, like genomic instability, there's issues because the repair proteins are mutated, they're not functioning correctly, but we don't want to get away from the fact that the regulated DNA damage is part of this. Cancer cells just don't have the aptitude to fix it appropriately. So let's talk deregulating cellular energetics. Stem cell metabolism is largely glycolytic, right? And actually, in a lot of ways, is similar to the Warburg effect. It's very normal, right? And at this point, there's a pretty good understanding of how, like, hexokinase 2, which is the cancer-associated form, links apoptotic regulation to energy metabolism. And when we recall that apoptosis is, of course, potentially differentiation signaling, that adds a little bit more, well, potential complexity to the interaction. We know, of course, that rapidly cycling cells, of course, need NADPH for biosynthetic processes, but also to maintain redox regulation systems like the glutathione and thyroxine ones I mentioned before. This is important, especially in stem cells, where that hypoxic condition seems to maintain stemness. HIF1 alpha, anyone? So let's talk resisting cell death, right? So death receptors and cell death mediators regulate differentiation in stem cells. It can even function as survival signals. So the inability to stabilize in G1 around a hypophosphorylated PRB might be restricting sufficient caspase activation to kill these cells. Though there is the caveat there that P21 has the ability to bind to, I believe, at least procaspase-3 and restrict the activation of this protease. So if we're seeing a situation where you have P53 or P21 mutated, or you're able to get to a hypophosphorylated PRB state without P53, you would still expect dysregulation of the system. It's just not overly clear to me at this moment exactly how that would work. So, tumor-promoting inflammation. This will be discussed. NF-kappa B is going to be discussed a lot in the second video. But NF-kappa B signaling is core to differentiation in a variety of contexts. As I kind of consider NF-kappa B signaling, I think of it as stress accommodation signaling, at least the canonical side. Not as familiar with the non-canonical side, though I've seen a couple of papers with Nick. Now, if we look at sustained proliferative signaling and evading growth suppression, now we've kind of covered how proliferation and differentiation are situationally complementary, but antagonistic when we're looking at the terminal differentiation process. And of course, you know, in cancer, loss of RB, loss of P53, loss of P21, that's going to complicate G1 arrest, and it might take us in looking at instances of DNA damage in preceding cell cycles to find alternate ways of getting these cells to arrest in G1, even without these proteins. I think there are ways. I just don't quite have them outlined at this moment. But kind of wrapping up here, because I expected this to be about 20 minutes. It's getting a bit long. I do not think it is either appropriate or correct to view cancer as hundreds of different diseases. It frustrates me. I think of hallmarks as signs of the disease. And if we're looking at all of these different types of cancer, and they all have the same symptomology, why would we assume that the underlying cause is different? I don't think it is. It really just comes down to aberrant differentiation, dysregulated differentiation, possibly, inappropriate activation, or more likely, resolution of a differentiation program. I think most tumor suppressors can really be thought of as protecting the fidelity of differentiation. And when we look at the signaling overlap between differentiation and death, that should allow us to reconcile some of these situational contradictions whereby tumor suppressors can have oncogenic potential in one context, but tumor suppressor functions in another. That duality of death and differentiation and the breakdown of that system is where that comes into play, at least in many cases in my mind. So if we appreciate how these systems interplay in like the healthy system, it'll be a lot easier for us to really understand what's going on in cancer, and I think find better targets. And in a lot of ways, this really simplifies things, right? All we have to do is really just figure out core mechanics of differentiation, bridge those to the defects in cancer, and identify inherent susceptibilities. I am personally looking for where the system breaks down. I don't know if there's one small or broad regulatory intersection, but there may be. And I am trying to figure this out. Cool. So, how are we going to be looking at these networks in future videos, right? How are we going to outline what is going on in these systems and where the important regulatory intersections are? And a lot of this is going to come down to an understanding that moonlighting proteins, or moonlighting functions of proteins, we do a really bad job in the field of biology, cell and molecular biology at the very least, of assuming that the first ascribed function of a protein is the most important. It may be, right? Like PRB, regulation of transcription, E2Fs, DP proteins, or transcription factors, like that's obviously going to be critical, but in a lot of other instances, I'm not so sure, right? And the issue with this is it gives people the perception that that is the one function of this protein. When you think about proteins that are multifunctional, what one really comes to mind? I mean, P53, right? What doesn't it regulate? But that degree of multifunctionality is, or a large degree of multifunctionality is ascribed to many of these other proteins. Like PRB can facilitate apoptosis at the mitochondria. What? Like that doesn't make sense. Or it's not something you would intuitively expect, but it has the potential to be broadly important. Cyclin D has a lot of functions outside of its interaction with cyclin-dependent kinases, but because we are taught that the function of cyclin D is facilitating a cell's progression through early stages of G1, we don't look at the interplay that could be going on in other phases of cell cycle, such as G2, as we mentioned above, where cyclin D upregulation in G2 allows for cells to exit mitosis in a environment that allows them to continue proliferating with a shortened G1 phase. Like even cyclin D interacts with VDAC to regulate its functionality at the automitochondrial membrane. So what we are going to do is kind of understand that even though when we look at the research, we get these snapshots of these proteins' functionality, we have to appreciate that a lot of them are going to be doing a variety of things in different phases of cell cycle, possibly sequentially, possibly simultaneously, and that the interplay of these interactions has to be understood in the context of the full system of a stem cell proliferating and becoming a terminally differentiated cell. When you actually look at a lot of these regulators of cell death, your caspases, your BCL2 proteins, your core apoptotic machinery, and you look at reviews on individual proteins, you kind of get the exact same thing. This protein was originally described for function A. However, it has subsequently been shown to be involved in proliferation, differentiation, death, energy regulation, and DNA damage responses. Those 5 are actually very, very common. They, of course, align with our understanding of what is required for stem cells to differentiate. They align with defects we see with the hallmarks of cancer. So what we're going to be doing is looking at how the multifunctionality of individual proteins can be utilized at different phases of cell cycle to coordinate this overarching program that encompasses proliferation, differentiation, DNA damage, and cell cycle. In doing this, it should give us a broader contextual understanding and allow us to bridge these diverse functionalities into a more cohesive whole. So, I think that that's probably enough for this video. This is a lot longer than I expected, and this was meant to be the basic one. Video 2, I think the PowerPoint is like 80 slides, so I might need to break that up a little bit, but I would very much welcome a discussion. I'm going to link a couple of the papers that I've read that I think are going to be really important down below. If you want to read them or you want to have conversations with it, please. Challenge my ideas, I encourage it, but really what I want is a dialogue here, because I want people to understand that a more nuanced understanding of differentiation is the fastest way to really understanding what is going on in cancer, and in my mind, finding more effective treatments. I know that there's no one in the world who's really looking for a cure. I'm not going to say that I am either. I'm just really trying to understand these systems, but it is so important that we make these cross connections here, because it makes it make so much sense, and it makes it so much easier to rationalize the behavior in the cells, and it really just brings it into perspective of not so much why is this happening, but how is it being mediated in a way that it allows for it to facilitate this overarching program as a whole. So if y'all are interested in death and differentiation as it relates to cancer, and want to immerse yourself in nuanced molecular mechanisms and regulatory control, hopefully you'll enjoy this channel. Please, let me know what you think. If you have any questions, let me know. And if you would like additional links to some of the information I would talk about, because I know it can be difficult to find them, to scour the literature, let me know. All right, cool, thank you.